Look closely, a thing so small you can't see it. Can it solve climate change, disease treatment, and other major global problems? Would you believe it? This is not a fantasy. The century-old problem that has plagued the scientific community, determining protein structure, has been conquered. And the power behind this might just be able to move the whole world. Think about the last 60 years. Scientists around the world have worked their tails off and only determined 150,000 protein structures, but a small team of a dozen or so people, in just a few years, has directly solved 200 million protein structures, covering almost all known proteins in the natural world. How did they do it? How was this achieved? Proteins, at first, are just a string of amino acids. The carbon atom in the middle of an amino acid is connected to an amino group on one side and a carboxyl group on the other. And it also has to connect to one of 20 different side chains. One of them, these amino acids form peptide bonds, linking into a chain. Under the influence of various intermolecular forces, this chain begins to curl and fold, forming a three-dimensional structure. This is a key characteristic of proteins. Take hemoglobin, for example. Its binding site is perfect. It's incredibly efficient at carrying oxygen in the blood. But in the past, determining protein structures was really slow and difficult. In the earliest days, to determine a protein structure, you had to crystallize the protein, then use X-rays to get a diffraction pattern, and then deduce the molecular shape. British biochemist John Kendrew spent a full 12 years to figure out the first protein structure, myoglobin. Over the next 20 years, only about 100 more structures were solved. Protein crystallization is difficult and expensive, often becoming the subject of doctoral dissertations. Why are proteins so complex? Scientists were also baffled and anxious. Determining the amino acid sequence costs only about $100. If we could use this to calculate how a protein folds, how much trouble would that save? Linus Pauling once predicted that the secondary structures of proteins are helices and sheets. Stumped everyone. Myth biologist Cyrus Leventhal calculated that a short chain of 35 amino acids has an absurdly large number of ways to fold. It would take a computer 200 times the age of the universe to find the correct structure. To solve this problem, in 1994, John Moult of the University of Maryland started the CASP competition with the goal of designing a computer model that could take an amino acid sequence and output a structure. At first, the results from the teams were abysmal. However, the Rosetta algorithm once led the way and even created the game Fold-It, allowing players to participate in protein structure prediction. Former international chess prodigy Demis Hassabis also participated. Later, Hassabis founded DeepMind and started the AlphaFold project. AlphaFold uses a deep neural network, inputting an amino acid sequence and evolutionary clues, and outputting a 2D pairing representation, which is then folded into a 3D structure by an algorithm. although it attracted a lot of attention when it participated in CASP13. Its score did not reach the 90-point standard. Hasabis brought in John Jumper and launched AlphaFold 2. This thing, in its deep learning system design, integrates geometric, physical, and evolutionary concepts. Accuracy immediately improved by a large margin, relying on supercomputing power, a large and diverse dataset, and more powerful AI algorithms. AlphaFold 2 achieved great success in CASP14. The predicted results were almost identical to the actual structures, exceeding the 90-point gold standard. With the success of AlphaFold 2, global research and laboratory work have been advanced by decades. It has helped develop a malaria vaccine, made it possible to break down antibiotic-resistant enzymes, and helped us understand the relationship between mutations and various diseases. His paper has been cited over 30,000 times. Jumper and Hasabis therefore received half of the 2024 Nobel Prize in Chemistry. The other half went to David Baker, who used generative AI to design completely new proteins, creating human-compatible antibodies that can neutralize deadly snake venom. AI's achievements in the field of proteins are just the tip of the iceberg of its potential. In other fields, such as materials science, AI is also making transformative leaps. It is pushing the boundaries of human knowledge at an unprecedented speed. In the future, it might bring even more shocking breakthroughs. With all the AI reports on the screen now, do you think strong artificial intelligence is still far away? Welcome everyone to leave a message and discuss.